Hello, so my name is Jeff Vassallo. I'm a Senior Director in Clinical Operations at Medpace. For those of you who don't know, Medpace is a clinical research organization. As all of you in this room are aware, cell and gene therapy trials are complex and labor-intensive, which is directly related to the large number of operational considerations which are part of the clinical development, which is in direct contrast to more traditional trials involving small molecules. This complexity is further magnified by the need to consider many operational activities early in clinical development to ensure studies are completed on time and within budget. Medpace has extensive operational experience in conducting global cell and gene therapy trials, and we've observed common trends that negatively impact the projected timeline and budget. So to better understand some of these trends, including the root cause, we critically assessed the performance of 20 recently conducted global clinical trials, including both autologous, allogeneic, phase 1 to phase 3, in addition to a variety of different therapeutic areas. Of these 20 trials, 17 were delayed due to issues in non-clinical development and CMC, clinical trial design, site and vendor feasibility, as well as regulatory authority submission. Collectively, these data demonstrate a common pattern of delays with a root cause associated with siloed decision-making early in clinical development up until first patient. The goal for today is to briefly highlight the patterns of siloed decision-making and associated operational outcomes contributing to delays in clinical development of cell and gene therapies. And perhaps more importantly, the goal is to discuss improved strategy, integrating cross-functional activities early in clinical development to enrich the likelihood of executing cell and gene therapy trials within the projected timeline and budget. This figure highlights the common trend in clinical development of cell and gene therapy trials, which is characterized by a process of siloed decision-making. Specifically, while the numerous stakeholders and accompanying processes are typically considered in clinical development, the decisions for the protocol, product design, in addition to operational nuances of clinical practice for the indication, are often considered independently and not thoughtfully integrated early in the clinical development process. Consequently, confirmation of suitability of the protocol as well as the product design occurs much later during regulatory submission and patient enrollment, thereby requiring rework, timeline extension, and an increased budget to complete the study. The next few slides capture examples of common trends of siloed decision-making that we have observed during clinical development, along with the negative outcomes. During non-clinical development, product design decisions are often not based on clinical data or clinical handling and logistics. Additionally, there is often a lack of consideration for the realities of patient care and standard clinical practice for the specific indication at both the country and site level. And together, the absence of thoughtful consideration for these operational parameters often results in a lack of compatibility with site process and a protocol which is difficult to operationalize. During the clinical trial design stage, protocols are often developed to include parameters which are not fully tested, scale-up and clinical feasibility processes for both obtaining target cell doses which are not fully vetted for the indication, global differences in logistics and clinical practice which have not been confirmed, and protocol-specific details dictated by the product design without ensuring compatibility with site process. Collectively, these decisions can lead to a protocol which is difficult to operationalize. Feasibility, qualification, selection, and startup of clinical sites and vendors often occurs after finalization of the protocol and product design. An unfortunate consequence of this late-stage activity is a protocol and a product design which may not be compatible with equipment, resources, processes, and logistics at both the country and site level, and thus require modification of both protocol and product design, which can lead to extensive delays. During the regulatory authority submission stage, changes to packaging, handling, labeling, and product logistics often occurs because there was no consideration for the global regulatory requirements during early clinical development. And the time needed to refine these product parameters and protocol design often leads to extensive delays. During enrollment, it is often discovered that manufacturing is not feasible with clinical samples and the realities of patient care. As an example, many protocols try to fit patient into a manufacturing slot instead of scheduling a manufacturing slot into patient care. While the operational issue is in part due to the current limitations of manufacturing, it is also often directly related to manufacturing feasibility conducted on non-clinical data or normal human volunteers. As noted in the beginning, we identified a common trend in clinical development of cell and gene therapies, which is characterized by a process of siloed decision-making. Specifically, while the numerous stakeholders and accompanying processes are typically considered in clinical development, the decisions for the protocol, product design, in addition to operational nuances of clinical practice for the indication, are often considered independently and not thoroughly integrated early in the clinical development program. Consequently, confirmation of suitability of the protocol as well as the protocol design occurs much later during regulatory submission or patient enrollment, thereby requiring rework, timeline delays, and increased budget. However, these data also suggest that implementing a strategy of integrated cross-functional activities early in clinical development can enrich the likelihood of success in executing these trials within the projected timeline and budget. More specifically, these data highlight the importance and value of integrating regulatory strategy with product and protocol design in early clinical development. As an example, it is important to ensure manufacturing of the cell product can be completed in the patient population instead of just normal human volunteers. Additionally, the protocol needs to be developed around the patient instead of the patient around the protocol. Logistics of operationalizing a global study with manufacturing in one country must be thoughtfully considered. Furthermore, the protocol and product design must be consistent with the unique regional differences and regulations. And finally, the consideration should be given for the global storage capabilities, product stability, packaging, preparation, and infusion to ensure compatibility to site process and clinical practice. Importantly, mock runs represent an excellent way to pressure test this process. Okay, and if you have interest in learning additional information about this topic, we are conducting a webinar next week that you're certainly welcome to join. Thank you for your attention.